inside the cell. And that organelle is the mitochondria. And I'm going to go ahead and draw the kind of two membranes that it has here. We're not going to talk about this too much, but just because it is important to remember that this has an inner membrane and an outer membrane. Remember that the electron transport chain is, of course, located on the inner membrane. And the mitochondria is also within it. It's the site of the Krebs cycle, which continues, notably, to break down glucose following glycolysis, which takes place inside of the cytoplasm. Now, since we ultimately want to get down to how extra glucose can be eventually converted into fatty acids, we need to actually make sure and remind ourselves how the breakdown of glucose proceeds. So as a very, very quick review, recall that glucose enters our cells from the bloodstream, and it enters the metabolic pathway called glycolysis, which takes place inside the cytoplasm. And the end product of glycolysis is pyruvate. And I'll also remind you that for every one molecule of glucose, which is a 6-carbon molecule, so 1, 2, 3, 4, 5, 6, we form 2 molecules of pyruvate, which is a 3-carbon compound. Subsequently, pyruvate is actively transported across the mitochondrial membrane by specialized carrier proteins located on the membrane, and once pyruvate reaches the inside of the mitochondria, also known as the inner mitochondrial matrix, there is an enzyme that's only found in the mitochondria called Pyruvate D Hydrogenase, often abbreviated as PDH, which oxidizes and removes 1 carbon from pyruvate. So remember, we had 3 carbons, and now it turns it into a 2-carbon molecule called Acetyl Coa. Now you might recall that this 2-carbon structure is not done being broken down or oxidized. There's still some energy that we can extract from this 2-carbon molecule, and it's extracted inside of the Krebs cycle. So remember that there are many, many intermediates along the Krebs cycle, but I only want to mention a couple that will be relevant when we talk about how this breakdown of glucose converges with the synthesis of fatty acids. So remember, first off, that a 4-carbon molecule called oxaloacetoacetate, which I'm abbreviating here as OAA, combines with 1 molecule of acetyl CoA to produce a 6-carbon molecule now called citrate. And citrate continues to be modified, oxidized, and even broken down a little bit more, and it returns to form oxaloacetate, which means that we lose 2 carbons somewhere along this cycle, which we do indeed. We lose these as 2 molecules of carbon dioxide. So there's 2 carbons of acetyl CoA exit as carbon dioxide, and we also form a number of reduced electron carrier molecules called NADH and FADH2, which shuttle their electrons from the oxidation process that occurs in the Krebs cycle to the electron transport chain, which is located on this inner mitochondrial membrane. How convenient, right? And then from there, we can produce ATP using oxidative phosphorylation. All right, so after that quick whirlwind tour of the breakdown of glucose, you might be wondering, where do we convert glucose into fatty acids? And it turns out that one of the intermediates of the breakdown of glucose, which is acetyl CoA, this 2-carbon molecule located in the mitochondrial inner matrix, is a precursor for fatty acid synthesis. And we're going to go through all of the steps, but just to take a step back for a moment, the big picture way that I kind of like to think about this is that remember that fatty acids, I'm going to go ahead and draw off to the side here. Remember that most of it is just a repeating carbon-hydrogen backbone, shown here in this kind of line stick model here. And so in that sense, really, we want to basically be able to link together carbon-carbon bonds. And this acetyl CoA is just a pair of carbon-carbon bonds that we can ultimately link together. Now, it turns out that we have an interesting situation when it comes to fatty acid synthesis and linking all of these acetyl CoA molecules together, which is that all of the enzymes necessary for fatty acid synthesis, I'm going to say enzymes for fatty acid synthesis, are located in the cytoplasm. And that's a bit problematic because remember, our acetyl CoA molecule is in the mitochondria. Now, your first thought might be, well, pyruvate was able to shuttle across using some protein carrier molecules in these membranes into the mitochondria, why can't acetyl-CoA do the same, going the opposite direction? Unfortunately, for some reason or the other, our body has evolved not to have any means to be able to transport this through its mitochondrial membrane. There are no protein transporters or carrier molecules like we had for pyruvate to be able to essentially shuttle acetyl-CoA in either direction across this mitochondrial membrane. But notably, our body does have a protein shuttle across this mitochondrial membrane for the molecule citrate. And remember that citrate contains acetyl-CoA. Of course, it also contains this molecule acetyl acetate that combines with it. And so let's see what happens when this shuttles across the mitochondrial membrane. Now, once citrate reaches the cytoplasm, it turns out that there is an enzyme within the cytoplasm that is able to break citrate up back into oxaloacetate, as well as the molecule that we're interested in, which is, of course, acetyl CoA. Now, when I first learned about this, it kind of struck me as a really roundabout way to kind of accomplish what seems like a pretty simple task, right? Which is to get acetyl-CoA into the cytoplasm where the enzymes for fatty acid synthesis can link it together to form a fatty acid. But it turns out that there might be a benefit for this citrate shuttle to make fatty acid synthesis perhaps more efficient. And so I want to briefly talk about that, but I want to erase this just to give us some more room. Now, it turns out that this 4-carbon molecule, oxaloacetate, is not going to be used for fatty acid synthesis. And so naturally, our body says, why don't we recycle it? And in fact, we do have some enzymes that can convert it back to this molecule, pyruvate. And notice that pyruvate can essentially, once it goes back to the mitochondria, it'll be turned into acetyl-CoA, and this entire cycle can continue. Now, although we're not going to go over the detailed mechanism by which oxaloacetate is converted to pyruvate, what is important, kind of a big picture idea to note here, is that we're going from a 4-carbon to 3-carbon molecule, and so we're going to lose a carbon actually as carbon dioxide during this process. And simultaneous with this step, we're actually oxidizing that particular intermediate. And so when we oxidize something, we're able to reduce something else. And it turns out that what we reduce in this case is a molecule of NAD+. And so it's reduced to NADPH. And you may recall that you've seen NADPH also as a product of the pentose phosphate pathway. And of course, we normally think about the pentose phosphate pathways being the major pathway for the production of NADPH, but this step also allows us to produce a molecule of NADPH as well. Now, one of the uses of NADPH that you might recall is that because it's associated with these electrons, it can serve as a source of reducing power to help with anabolic reactions. And remember that anabolic reactions are anything that involve building up a molecule, including fatty acid synthesis, which is exactly what we're trying to accomplish here. So to summarize and just kind of tie up everything that we've just talked about here, we've been able to get acetyl-CoA into the cytoplasm where all of the enzymes necessary for fatty acid synthesis are located. And this is important because we're going to use acetyl-CoA, multiple acetyl-CoA kind of a precursor molecule, so to say, to build up a fatty acid. And of course, because this is an anabolic reaction, we're going to need some ATP somewhere along the way. And we're also going to need some reducing power to kind of help form all of those carbon-carbon bonds. And we can get that using NADPH. Of course, NADPH can be supplied by the pentose phosphate pathway, but conveniently perhaps, by using this citrate shuttle, we're also able to produce a molecule of NADPH from the conversion of oxaloacetate into pyruvate. In the next video, we'll pick up right here in the cytoplasm to talk about how this conversion from acetyl-CoA into a fatty acid occurs.
Okay, so any questions so far before we continue to the second video? Everyone okay? You guys understand? Can we continue?
Yes, yes.
Okay, so if you don't understand, you can always refer back to this video on YouTube. Okay.
To return to some basic principles and to put everything in perspective for you, I want to remind you that fatty acid synthesis is like any other anabolic reaction that occurs in our body. Remember that the common theme with anabolic reactions is that we take some type of monomer, generally, and we link these together to form a polymer. And the challenge with these types of reactions is, of course, that generally these reactions have a positive delta G value, or in other words, they are not spontaneous. They require an input of energy to occur. So obviously this is a challenge, but our body resolves this challenge by coupling this reaction with a reaction that has a favorable delta G value, that is a delta G value less than 0. And in this case, our body uses the energy currency of our cell, ATP. And of course, we know its reaction with water has a very favorable delta G value, very negative. And so when we break it down, we form ADP and a free phosphate group. Now, I just want to remind you here that our monomer, when we're talking about fatty acid synthesis here, is Acetyl Coa. So these two carbon subunits, we're going to link these together to form a large fatty acid chain. Now, before we get into the specific steps of fatty acid synthesis, I want to kind of take a bird's eye view first and write out what the overall chemical reaction would be for fatty acid synthesis occurring in our body. So essentially what I'm doing is I'm adding these two equations together. And just to make this super clear, I want to remind you that in order for this reaction to occur, obviously we need to have a favorable delta G value. So overall, this is going to have a negative delta G value, which tells you, of course, that the hydrolysis of ATP is much more negative than this polymerization reaction is positive. So, of course, if we add these two delta G values together, we get an overall negative delta G value. All right, so the overall chemical reaction that I want to write out for fatty acid synthesis is for the product of a 16 carbon fatty acid chain that is called palmitic acid. And palmitic acid happens to be the primary product of fatty acid synthesis in our body. So we're going to focus on this, but just so you know, our body can essentially use this kind of basic product of fatty acid synthesis to synthesize longer chain fatty acids if need be. So remember that we want to start off with our monomer, which is Acetyl Coa. And I'll remind you that this is a 2-carbon molecule. So if we need to make a 16-carbon fatty acid, we would need 8 acetyl CoA molecules. And of course, since this is an anabolic reaction, we need ATP to make an appearance as well. And it turns out 7 ATP molecules are required for this reaction. And in addition, we also actually use a molecule called NADPH. And you might recognize this from the pentose phosphate pathway, but remember that we've also produced NADPH when we've shuttled this acetyl CoA over to the cytoplasm from the mitochondria, because remember that was done by a molecule called citrate, and citrate broke up into acetyl CoA as well as oxaloacetate, which the oxaloacetate produced this NADPH as it was being recycled back to pyruvate. And this is really important because it's a source of electrons that will be able to reduce any oxidized groups, any groups that are oxidized on this acetyl CoA molecule. So what I will show you in a minute is that acetyl CoA has a carbonyl group in its molecule. And of course, we want to make just carbon-carbon bonds that are essentially attached to hydrogen only. And so in order to reduce this bond, we need a source of reducing power, and NADPH is that source of reducing power. And it turns out that we need 14 NADPH molecules. So that's all of the reactants that we require, and we form this palmitic acid, and of course we also form the byproducts of the rest of these reactants. So we form, if we started off with 7 ATP, we end up with 7 ADP, and of course 7 free phosphate groups, and 14 NADP plus, because we've removed these electrons, and of course we're oxidizing this now, and we also end up losing some water molecules as well during the course of this reaction. End up losing 6 water molecules. And I'll actually go ahead and move the 14 down here so it doesn't confuse you. And I also forgot just one more thing here. So it turns out that these coenzyme A functional groups here on this acetyl-CoA molecule will also be removed in the process of making these carbon-carbon double bonds. And so will also form as a byproduct 8 coenzyme A molecules. All right, so I'm going to go ahead and scroll down so that we can return to the cytoplasm and figure out how acetyl-CoA is polymerized into this palmitic acid. Now, because I know that it's quite easy to get kind of lost in the details, especially when talking about metabolic pathways, my goal here is not to provide kind of a detailed reaction mechanism for every step of this reaction, but rather to kind of just cover the high yield steps, the steps that are kind of the most important, I think, for you to be able to take away. All right, so once we're in the cytoplasm and we have acetyl-CoA at our disposal, turns out that the first step of fatty acid synthesis is to charge up this molecule to a higher energy molecule called malonyl CoA. And just for the fact that I'm saying charging up this molecule should clue you into the fact that this is thermodynamically unfavorable, and so we need something that is thermodynamically favorable to be coupled with this reaction, and indeed that is the hydrolysis of ATP, which I'll just write shorthand here as ATP going to ADP plus a free phosphate group. Now, it might be tempting to think that we're charging up this acetyl-CoA molecule with this phosphate group here, but what we're really doing actually is we're charging it up with a carbon dioxide molecule. And the breakdown, this hydrolysis of ATP, is really what's fueling this reaction forward. So to give you a sense of what kind of this looks like chemically, let me go ahead and draw out the chemical structure of these two molecules. So acetyl-CoA, we begin with this kind of 2-carbon backbone structure that we call an acyl group. So carbon double bonded to oxygen and also bonded to this carbon CH3 group here, is attached to a coenzyme A group via a sulfur atom here, and I'm going to leave this CoA group abbreviated like this just for simplicity. And our malonyl CoA group looks very similar. So we have a carbon double bond to oxygen. Our coenzyme A group is untouched, but we end up, plopping off a hydrogen here and essentially adding on this carbon dioxide molecule like such. And I'll go ahead and put a hydrogen here just for now, but it might be protonated or it might not be, depending on the pH of the solution, obviously. But just to kind of give you a sense here, this is what malonyl CoA looks like. Now, after this activation step, we're ready to polymerize. We're ready to put these malonyl CoA molecules together to form the 16-carbon palmitic fatty acid. So to do this, it turns out that this carbon dioxide molecule that we added to this malonyl CoA group, it plops off once again. So it might kind of seem weird to you that we're adding a carbon dioxide molecule and it plops off again, but what this is really doing, as I'll touch on in a bit, is it's making this overall reaction thermodynamically favorable, because otherwise it wouldn't be able to occur. And notably, we also happen to lose some water molecules as well in the process. And this is also where the addition of NADPH comes in, because we need the NADPH to reduce these carbon double bonds, as I mentioned earlier, so that we can create that nice string of carbon-carbon and carbon-hydrogen bonds that is what forms the bulk of our fatty acid. Now, I do want to get the stoichiometry right here and remind you that what I've written out here, this loss of one water molecule and this loss of the carbon dioxide group and the addition of these two NADPH molecules, is for each subsequent addition of these 2 carbon subunits. So every time we make another cycle, we're going to have another 2 NADPH and another loss of carbon dioxide and another loss of water. And that will essentially continue until we've built this 16-carbon palmitic acid chain. Before I touch more on this polymerization, let me just actually scroll down here and talk a little bit about the enzymes that are carrying out these kind of two major steps. So the first enzyme that carries out this activation step has a very nice name that's easy to remember. It's called Acetyl Coa Carboxylase. So essentially tells us exactly what it's doing. It's adding a carboxy group to this acetyl CoA molecule. And what's important about this particular enzyme, let me scroll down a little bit more here, is that it ends up being a very highly regulated part of this entire sequence. Now, the reason this is a really good step to regulate is because it happens to be the rate limiting step of this entire fatty acid synthesis pathway. In other words, out of all the chemical reactions that occur, this one is the slowest kinetically. And so, because the entire rate, the overall rate, how fast this fatty acid synthesis pathway can occur is limited by this one step, it makes sense that we should essentially regulate whether this step is on or off. And the way we do this is by allosteric regulation, which, if you remember, is a specific molecule binding to a non-active site portion of this enzyme to make it work better or worse, or can also regulate it with hormones as well. So I'm going to say hormonal. So I just want to mention a couple of these that are going to be relevant. So first off, we have citrate, which is an allosteric activator of this enzyme. It makes this enzyme work better, and hence increases the flow through this pathway. And this kind of makes sense to me because I think of the citrate shuttle that's shuttling all of this acetyl CoA to the cytoplasm. More acetyl CoA means that we should take advantage of this acetyl CoA and make more fatty acids, hence kind of moving this pathway forward. In terms of hormones, the hormone that activates this pathway is insulin. And this makes sense to me again because I know that right after, chowing down on that cheeseburger, we know that our levels of glucose in our blood are also going to rise. And so, if we have a lot of extra glucose around, we're going to have a lot of extra acetyl CoA. Hence, once again, we want to move this acetyl CoA down this pathway to produce more fatty acids. Now, in terms of inhibitors, allosterically, it turns out that fatty acids, especially I'll say long-chain fatty acids, can inhibit this enzyme. And the way I like to think about this is it's just kind of a form of product inhibition. If we have too many fatty acids being produced, this process is basically the cell is saying, hey, acetyl CoA carboxylase, you can slow down a little bit. We have more than we need. And in terms of hormones, the hormone that inhibits this enzyme is glucagon, as well as a couple of others, but this is one of the main ones. And the way I like to think about this is kind of twofold. So remember that glucagon levels rise several hours after a meal once we have low blood glucose levels. So if we don't have an excess of glucose, we're not going to have an excess of acetyl CoA, and so we won't want to be, forming all of these fatty acids. But I also remember that glucagon, the rising levels of glucagon also signal those adipose cells to release those fatty acids into the bloodstream for all of our cells to break down to produce more ATP. So if we're in a state of kind of net breaking down of fatty acids, we don't want to be simultaneously building them back up, because ultimately we want to be able to, in this phase at least, extract the energy by breaking those fatty acids down. Now I want to mention what the enzyme is called that polymerizes these malonyl CoA subunits together to form this palmitic acid. And the name of this enzyme is also quite easy to remember. It's called fatty acid synthase. So we're synthesizing a fatty acid. Now, as you probably saw from this arrow, there's a lot of things going on here. We're adding NADPH molecules, a carbon dioxide molecule is falling off, and water is falling off too. And we're not going to go over all of the reactions that occur in this enzyme, but I think if we scroll down here a little bit, it'll be helpful to kind of just get a sense for how these carbon-carbon bonds are forming. Now, to save us some time, I've pre-drawn a couple of things that I want to go ahead and explain to you right now. So starting here on the left, what I've drawn here is the fatty acid synthase enzyme. And what I want to kind of draw your attention to, obviously this isn't what the enzyme actually looks like, but a representative drawing, is the fact that it has two identical subunits shown here, and they each have a thiol group or a sulfur hydrogen group located here in the active site region. So the very first step of this polymerization reaction is actually to start off with both a molecule of acetyl CoA and malonyl CoA. And one acetyl CoA molecule attaches to one of those sulfur groups, and the malonyl CoA molecule attaches to the other sulfur group so that they're in close vicinity of one another. But I do want to point out that obviously these are no longer called acetyl CoA and malonyl CoA because you can notice here that there's no coenzyme A anywhere. And indeed, when these carbons here formed a bond with the sulfur atom on these enzymes, they lost their coenzyme A group. So that's kind of where we lose our coenzyme A group right here. Now, in terms of carbon-carbon bond formation, I want us to put on our organic chemistry hats on for a moment. Now, I've already told you, up above here when we were looking at this reaction mechanism, that we lose this carbon dioxide molecule that we've added in this malonyl CoA molecule. But how does this impact the carbon-carbon bond formation? So here I want you to notice that when this decarboxylates, when these electrons decide, hey, let me form a double bond here and let me kind of plop off of this molecule, that these electrons now make this carbon very nucleophilic. In other words, these two electrons want to bond with something. And so what they'll do is they'll see the nearest carbon that has a suitable leaving group around, and it happens to be this carbon right here because this enzyme is nicely putting us in vicinity of another carbon bond. And when it makes this carbon bond, this carbon will say, hey, I like that bond a little bit better. I'll plop off and give you my extra electrons. So what we've done essentially is we've made a carbon-carbon bond and we've freed a space here to allow the input of another malonyl CoA molecule. And this cycle will continue to repeat, us making carbon-carbon bonds until we form the 16-carbon palmitic acid. Now, I know we haven't really covered where the NADPH comes in to reduce these carbons bonded to oxygen or where there's a loss of water, but I will actually mention that it's quite similar to the reverse of beta oxidation, which you can take a look at as well. But ultimately, after all of that is done, after we make this carbon bond, which I think is the most crucial part of this fatty acid synthesis, and we reduce those carbons bonded to oxygens, ultimately we form this 16-carbon palmitic acid, which I've drawn the structure of below. So it's a completely saturated fatty acid that we can use to make longer chain fatty acids if need be. And I want to remind you that the ultimate fate of these fatty acids, remember, they're going to be attached to a glycerol backbone to form a triacylglyceride molecule, and those are going to be sent off by the liver via VLDL particles to essentially deliver these fats to the rest of our tissues.
Okay, any questions so far? Fatin, are you okay?
Okay.
If I ask you to summarize, can you summarize this process? Or you need to watch again? Adiba, are you okay? Everybody seems awfully quiet, means either you understand very much or you understand nothing. All right. But as I said to you, you can always watch again, and I hope eventually you will understand because everything that we learned so far actually connected to each other, especially the first video. It shows you guys how the breakdown of the glucose happens in the liver. And then if we have excess glucose, means too much glucose, and of course we have the insulin release and so on, we will produce a lot of acetyl CoA. So this acetyl CoA will enter into TCA cycle and form citrate first before the citrate can be shuttled into the cytoplasm of the liver cell. So most of the fatty acid synthesis happens in the liver cell, and also it happens when we have a lot of glucose. Okay, so once the citrate enter into the cytoplasm, the citrate will be break back into two components, which are, the first one is oxaloacetate, and the second one is acetyl CoA. So this oxaloacetate will reform the pyruvate, and this pyruvate can enter back into the mitochondria to complete the cycle. And you also notice one new molecule, which is NADPH. Okay, the one that we learned before this is NADH without P. So the main difference is NADH is for the catabolism breakdown, but NADPH is for anabolic reaction, okay, or building up. So the one that form oxaloate acetate is not very important for us to remember or to understand, but the important one is the formation of acetyl CoA from citrate. Okay, this one, acetyl CoA, because this acetyl CoA will be used to form the palmitic acid. So when we learned the beta oxidation last week, we learned that one palmitic acid can form many acetyl CoA. So the exact number is 8 lah. So if we have more or longer chain of fatty acid, we will produce more acetyl CoA. So contoh kalau kita ada 20 carbon fatty acid, we will produce 10 acetyl CoA lah. We just divide by 2. But the beta oxidation need to be minus by 1. So you will notice this one as well in this week, masa kita belajar ni. We will require, for example, 8 acetyl CoA, but the ATP or NADPH is not exactly the same as acetyl CoA. So ATP yang kita perlukan adalah 7, bukannya 8, sebab sama proses macam beta oxidation, dia kena tolak 1. And NADPH requires 14 NADPH, all of them, because one process requires 2. So 7 times 2 equivalent to 14 lah. And of course you produce some water as well along the way. Okay. Ada apa-apa soalan? Okay, this is the
so yang minggu lepas kan dia from the fatty acid, lepas tu dia ada chain reaction until dia produce acetyl CoA tu kan, makin kecil tu kan. Tapi yang tadi tu dia from acetyl CoA dia jadi malonyl tu until dia jadi acid balik.
Betul. Dia terbaliklah proses dia. Last week yang kita belajar pemecahan, the breakdown of fatty acid into acetyl CoA. But this week it's reverse from acetyl CoA to fatty acid. Ini terjadi bila kita from apa?
Okay. Tak, so from acetyl CoA tu, from acetyl CoA yang 2 carbon tu, dia akan end up akan jadi yang 16 carbon lah.
Betul. So maksudnya tadi kan kita dalam acetyl CoA tu, acetyl CoA kan 2 carbon kan?
Ya.
So kalau kita nak hasilkan 16 carbon palmitic acid, berapa acetyl CoA kita perlukan?
8
lah kan? So kita perlukan 8. Dia sebenarnya proses dia agak macam mana? Dia macam ada detour sikit tau. Dia tak exactly acetyl CoA terus jadi fatty acid. Kalau pasal tadi dia ada form malonyl CoA first. Okay? So saya rasa ada orang yang confuse sikit lah dekat malonyl CoA tu. Siapa yang confuse dekat malonyl CoA tu?
Saya.
Okay, saya. So kita tunjuk sikit lah. Okay, saya tak suka go into the chemistry lah. So kita cerita macam kita baca buku cerita je. Alright. So kita tahu acetyl CoA 2 carbon, betul tak? Can you see my drawing?
Yes.
Okay. So kita akan form malonyl CoA. Kita senang nak cakap malonyl CoA is a 3 carbon molecule. Okay, understand? Okay, ni acetyl CoA. Ni malonyl CoA. Faham tak?
Faham.
Okay, acetyl 2, malonyl 3. So in order for us to add 1 carbon, what we need to do is we need to add 1 CO2, betul tak? Nak tambah 1 carbon. Alright. So like the video just now, it says that the name of the enzyme is Acetyl Coa Carboxylase because it adds 1 carbon dioxide to the molecule. Kalau kita buang 1 carbon, kita panggil apa? Okay, kalau tambah kita panggil carboxylase. Kalau buang 1 carbon, kita panggil decarboxylase lah, betul tak?
Betul.
Okay, so saya nak bagi tahu je lah kalau korang ada nampak certain molecule that reduce in carbon number, for example from 4 carbon becomes 3 carbon, means that dia guna decarboxylase lah. Tapi kalau tambah, kita panggil dia as carboxylase. Okay, so in this case, nama dia acetyl CoA Carboxylase. And you need to remember this acetyl CoA carboxylase is a very important enzyme sebab ni yang kita panggil as regulated points. Okay, dia perlu ke ATP juga lah sini. Macam regulated points yang kita belajar dalam glycolysis kan, ada 3 kan, reaction 1, 3 and 10. So in the fatty acid synthesis, ni regulated points. Okay, the purpose of adding 1 carbon to acetyl CoA to form malonyl CoA here, the main purpose is later on dia akan potong balik carbon ni untuk drive the reaction forward. Sebab reaction ni dia tak favorable tau, dia anabolic reaction, so dia perlukan something energy untuk membolehkan reaction tu berjalan ke depan. Okay? In this case, dia akan potong balik 1 carbon daripada sini dan dia akan jadi 2 carbon balik. Saya tunjuk gambar sikit. Satu ramai agak confusion kat sini. So faham tak dia jadi 3 carbon tu?
Saya terfikir juga kenapa daripada acetyl CoA tu dia nak jadikan malonyl tu.
Okay.
Kalau dia tambah 1 tu, apa jadi dekat 16 tu? Macam mana boleh 16 balik?
Tu sebenarnya Tuhan punya kerja lah semua ni kan. Tapi based on biochemistry, saya cuba terangkan lah. Okay, ni contoh gambar, nampak tak? Okay, acetyl CoA becomes mana CoA kan, 3 carbon kan? Okay, all these things happens on fatty acid synthase. Ingat tak nama enzyme ni from the video just now? Ingat tak fatty acid synthase tu? Ni.
Ingat.
Okay. Kan dia cakap daripada acetyl CoA becomes malonyl CoA, and then from malonyl CoA dia akan undergo fatty acid synthase and form 16 carbon palmitic acid. Okay. So I just show you in detail lah what happens. So what happens is the first molecule that will come and attach to the fatty acid synthase is acetyl CoA. Only the first molecule. So apa yang jadi dia datang dia akan attach dekat sini. So CoA akan keluar lah sebab dia dah attach dekat salah satu active site on fatty acid synthase. So understand this first figure? You understand?
Yeah.
Okay. Then what happen is this molecule, the acetyl CoA just now, it will move to the second one, which is this one. Okay, faham tak so far yang tu?
Faham.
Okay. So when it comes to this one, this area will be available, right? Will be empty, right? Area ni. So what happen is bila dia empty, malonyl CoA yang kita hasilkan tadi, it will come and bind to here. Understand tak yang ni?
Faham.
Okay. So kita pergi next slide. Nanti saya upload. Okay. Kan tadi we just add 1 carbon here. 3 carbon kan malonyl CoA kan?
Yes.
Okay. So in order for this molecule to combine together here, they need to get rid of 1 carbon dioxide that we add just now. So bila potong tu dia hasilkan energi untuk menggabungkan dua molekul ni. So daripada atas ni dia akan turun bawah, dia akan gabung kat sini. Faham tak?
Faham.
Okay. So bila dah bawah ni jadi 4, dia akan naik atas balik. Jadi 4 dekat sini. So ini available balik kan? Malonyl CoA boleh masuk dan bind kat sini balik. Faham tak so far sampai sini?
Faham. So
Okay.
yang merah tu dia akan lekat dekat yang cuma 2 tu je lah kan?
The merah one will be, dia akan putus, dia akan keluar untuk menghasilkan, macam ATP kan dia boleh potong 1-phosphate to produce energy kan. Dalam kes ni malonyl CoA dia boleh potong satu ni, dia keluar kat dia boleh menyebabkan 2 ni condense, bergabung. So nanti bila dia potong ni, yang ni akan turun bawah, dia akan melekat, dia akan gabung sampai jadi 6 pula. Understand up to here?
Faham.
Okay. So 6 ni apa akan jadi? Dia akan naik atas balik, betul tak?
Betul.
Dan ni available balik kan?
Betul.
So apa yang jadikan available ni?
Melanin CoA.
Melanin
Datang
will
balik.
come and bind. Okay, and the same process repeats again. So altogether there are 7 reactions. You don't have to remember lah 7 ni. Tapi yang penting saya tunjuk the 7th reaction, the last one which is thioester, it only happens once. Kan reaction tadi jadi 7 kali kan untuk hasilkan 16 molekul. Reason dia 7 kali itu sebab sama macam beta oxidation lah, dia gabung sampai 7 kali. Tapi yang last ni, 7th reaction ni jadi sekali je. Sebab bila dah 16 baru reaction yang terakhir ni jadi untuk potong 16 karbon ni daripada fatty acid synthase. You understand tak kenapa yang last reaction ni jadi sekali je? Kira confused atau? Okay, semua proses kat atas ni dia jadi berulang-ulang kali sebanyak 7 kali. It happens 7 times. But only for the 7th reaction, it happens once only in all the reaction. This enzyme, thioester. Siapa tak faham kenapa dia jadi sekali je?
Maksudnya dia dah berhenti kat situ lah. Berhenti dah.
Maksudnya bila dah form 16 karbon tu, baru reaction dia jadi untuk potong fatty acid ni daripada fatty acid. Tu dia tak jadi 7 kali, dia jadi sekali je.
Doktor. Saya macam tak boleh nak faham je.
Okey, nanti tengok. Sebenarnya benda ni awak tak perlu tahu in detail, tapi saya tunjuk ni supaya awak faham semua proses yang terjadi tu. Kan macam kalau kita faham benda yang complicated, kita boleh faham benda yang lagi simple. Faham tak? Contoh tadi kan macam video tu dia tunjuk 2 karbon jadi 3 karbon kan. Lepas tu daripada 3 karbon ni dia tunjuk ada fatty acid synthase, tiba-tiba dah jadi 16 karbon kat sini. Yang saya tunjuk ni apa yang jadi in detail dekat sini. Dia tak tunjuk ni sebab jadi complicated lah. Kalau saya tunjuk, kalau korang faham ni, korang faham lah kenapa dia jadi 16 karbon tu. Tapi tak apa, kalau tak faham nanti maybe boleh datang singgah. Tapi saya dengar ramai kena COVID recently. Saya boleh tunjuk in writing lah, lagi senang nak faham lah. Okay, so daripada situ kalau siapa yang faham, actually you can build the overall reaction. Okay, just now we understand that we need 8 acetyl CoA, right? Betul tak? Plus 7 ATP plus 14 and ADPH. Okay, so this is the basic one. Okay, but in reality, actually, what happens to this 8 acetyl CoA is 1 acetyl CoA plus 7 malonyl CoA. Siapa yang faham kenapa dia macam ni sebenarnya?
Faham.
Okay. Dia jadi 8 acetyl CoA because 7 of this acetyl CoA actually being converted to malonyl CoA. Kan kita tengok tadi reaction tadi, only 1 acetyl CoA is being used on fatty acid synthase. Dia macam sumbu yang untuk start the reaction. But after that, all the subsequent reaction all using malonyl CoA, not acetyl CoA anymore. So in reality, actually, 1 acetyl CoA plus 7 malonyl CoA plus 7 ATP and plus 14 and ADPH. That is more accurate. But if you write 8 acetyl CoA, is correct as well. Okay, saya tunjuklah overall reaction dia sini. Okay, boleh tengok slide ni. This one macam ni. 1 acetyl CoA plus 7 malonyl CoA. Okay, equivalent to 8 acetyl CoA lah. Sebab actually this malonyl CoA also comes from acetyl CoA. Okay, so this is just the last slide. I just want to show you guys if the body needs the primary fatty acid that we produce is palmitate lah, 16 carbon. If the body needs shorter fatty acid, the thioester will come and cut at a shorter amount. Contohnya kalau diperlukan 10 carbon fatty acid, dia akan potong kat 10 lah. Tapi kalau dia perlukan longer fatty acid than 16 carbon, the liver cell will still produce palmitate. But this palmitate will be transferred to other component of the cell to be elongated at a different organelle. So kalau tahu difference yang tu je lah. but it's not that important lah for you to know. So far ada soalan lagi tak? Do you have any questions so far? Lagi? Can write in the chat if you are too shy to ask. Ataupun later on boleh study lagi. Okay, everyone. Azlin, Nick, Izzati, ramai ni. Jihan. Kata ada soalan, nak take 5 ke nak continue to protein?
Take 5.
Take 5.
Take 5. Okay, semua dah berasap
Yeah.
Okay, so we take 5. So after this, we will enter into the nitrogen metabolism. So if you know nitrogen is part of protein lah, so it's actually protein metabolism. Sekarang kita dah belajar glucose, fatty acid, and next protein. Protein ni maybe kita belajar sikit sahaja. I think we can skip protein synthesis lah, tapi kita boleh bincang later on. Okay, so we take 5 before we enter protein metabolism. Thank you.
Tapi dari sini tak akan bersih sebab jenggot tu besar.
Okay, can we continue?
Yes.
Boleh.
So nak belajar ke tak protein synthesis? Ke nak belajar metabolism saja?
Kalau tak belajar tak masuk exam doktor.
Tak masuklah. Kalau masuk nanti menangis pula kan korang tengah exam. So ni kita tak belajar lah. Okay. Sepatutnya kena belajar tau. Tapi ni saya offer. Tak perlu belajar tak apa tak. Sebab tak sempat lah nak masuk ni. Slide nombor 34 ni.
Saya okey je doktor.
Kebanyakan saya rasa okey je lah. Tapi jangan report dekat BHAP pula. Okay. So we will enter into nitrogen metabolism. As you can see here, it means that this will involve protein lah. Okay. Because nitrogen is one of the essential element in the amino acid. As an introduction, protein also can be used as an energy source, but the body will always prefer to use glucose first as the primary energy source. Okay, as you can see from this table, glycogen, there are two sites of glycogen. The first one is in liver and the second one is in muscle. In muscle, there is more glycogen because muscle will always need energy to move lah, to do all the movement in the body. And the glycogen in liver is actually lower, but it is important to stabilize the blood glucose level. Okay, so glycogen yang ada dalam muscle ni is for its own usage, but the glycogen that is in liver is for other usage, especially to stabilize the glucose in the blood. So liver ni kira unselfish lah, dia tolong stabilize yang lain. Tapi muscle ni selfish, dia guna untuk diri sendiri sahaja. And as you can see from here, the energy from 70 grams of glycogen is 280. So if you divide 280 by 70, you will get 4. So maksudnya 1 gram glycogen or glucose is equivalent to 4 kilocalories. Tapi kalau tengok triacylglycerol, which is fat in adipose tissue, it will produce up to 135,000 kilocalories because and if you divide this one 135,000 divide by 15, you will get about 8 to 9 kilocalories per gram. So maksudnya fat dia menghasilkan lebih banyak tenaga daripada glucose. Okay, that one we also learn in the fat metabolism last week lah. Fat will always produce more energy than glucose. And if you can see from here, protein in the muscle, 6,000 grams will produce about 24,000 kilocalories. So if you divide this one, you will get 4 as well. So protein menghasilkan sama banyak tenaga macam glucose. Tapi fat menghasilkan tenaga yang paling banyak sekali. Okay, in human, we always excrete the extra protein that we have. Okay, so for example, if you take a lot of protein and so on, you will probably urinate more, okay, pergi ke toilet lebih because there will be a lot of nitrogen that need to be excreted out from your system. Okay, in human being, the major type of the nitrogen excretion is urea, okay, which is about 86%. Tapi kalau kita tengok other organism, for example, fish and reptile, they excrete nitrogen as well, but in a different form. So fish will usually excrete the nitrogen in the form of ammonia or just one nitrogen, but reptile usually excrete the nitrogen in the form of uric acid, okay, which is up to 4 nitrogen. And we actually also excrete this one, but in a smaller amount. For example, ammonium, we just excrete about 2.8%. Uric acid is about 2 to 3%. So if we have imbalance in this one, it can lead to diseases. For example, orang yang banyak uric acid in the system, they can experience a disease we call as gout. Usually gout ni happens in obese people, older people and so on, when their metabolism is becoming imbalanced. So just for more introduction, protein biosynthesis will require amino acid as the monomer, the smallest part. And the source of amino acid can come from food, second from the degradation of protein within the cell, and the third one is synthesis from the metabolic intermediates. The synthesis from metabolic intermediates is the intermediates that we can see in the Krebs cycle or TCA cycle. Cellular protein are constantly being degraded and resynthesized. The short-lived protein usually play important metabolic roles. Okay, ni especially important for the protein that is used for signaling and so on. So kan bila kita sentuh something, kita rasa something, and then kita hilang sensation tu because this protein is constantly being degraded and resynthesized depending on the usage at that time. So this is the metabolic relationship of amino acid. As you can see from here, this amino acid can form the acetyl CoA or it can form the carbohydrate intermediates in the TCA cycle. And you can also see actually this being represented in different color as well. Yang ni kuning, yang ni merah. Okay, this one is red, this one is yellow. Just to show you guys that if the amino acid can produce acetyl CoA, we call this one as ketogenic amino acid. Okay, equivalent to ketone bodies yang kita belajar last week. But if it can produce other than acetyl CoA, the other intermediates in the TCA cycle shown in red, kita panggil dia as glucogenic amino acid, something that can produce glucose. So kena ingat dua ni penting sebab amino acid ada banyak kan. There are about 20 major amino acid, okay, that can be coded in the protein. For example, alanine, glutamate and so on. Ada setengah yang boleh produce acetyl CoA and ada setengah yang tak produce acetyl CoA. Yang tak produce acetyl CoA, dia boleh eventually produce glucose. Tapi yang tak boleh produce glucose, eventually dia akan produce ketone bodies, okay, which is part of the fat metabolism. So ada dua benda nanti kita tengok in detail yang ni. Yang ni kita boleh skip, not very important. Okay, any questions so far? Ada soalan tak?
No.
Okay. So kita enter into the degradation of cellular protein or proteolysis. So the degradation, kan kita cakap protein ni always being resynthesized or always being degraded depending on the usage of the body. So the degradation of this protein can occur based on two mechanisms. The first one is lysosome, non-specific or ubiquitination. So lysosome ni dia akan degrade je any protein, but ubiquitination ni lagi specific. I think probably you learn this one in your matriculation. So we will take a look at the specific one, but not the lysosome. So what happens is when the protein is broken or destined to be broken or is malfunction or is very old, what happens? The body will put certain kind of tag. Okay, ni tag baju ni, to show to the body that this protein is destined to be degraded. Okay, so this process we call as ubiquitination. It involves three enzymes, which is E1, E2, and E3. So after it attach this tag to the protein that when want to be degraded, it will be degraded by one enzyme called as proteasome. Okay, it will break this protein into smaller part that can be reused or secreted in the urine. Okay, it will be reduced to 7 to 9 residues. So this is the process. Ni protein yang kita nak degraded. There will be three enzymes that will come and attach this ubiquitin tag. Okay, so this ubiquitin tag can be thousands long or 100 ubiquitin long. Bila dia dah siap attach, then it will be recognized by this proteasome, this big blue and purple protein, and it will chew or eat this protein into smaller peptide fragments that can be reused or excreted. So this is the process in detail. So what happens is the E1 or enzyme 1 will activate this ubiquitin tag, and then this ubiquitin tag will be released from enzyme 1 and bind to enzyme 2. Okay, dia akan bind to enzyme 2. And lastly, enzyme 3 will come and bind to the substrate that we want to put the ubiquitin in, and it will actually act as bridge for this ubiquitin to be transferred to the substrate. And then this process will happens over and over again until we have a long chain of ubiquitin. Once it reaches the required amount of ubiquitin, it will be degraded by proteasome. There will be many videos as well in the YouTube about this process if you want to know in detail. So what is the fate of this amino acid that is being degraded just now? So either it will be degraded by the amination of amino acid, or it will be reutilized into different amino acid or alpha keto acid. The alpha keto acids from the amination can be metabolized as precursor for other reaction, for example, gluconeogenesis, ketogenesis, or as TCA cycle intermediates. So alpha keto acid is actually the chemical that we can find in glycolysis, TCA cycle, and so on. Later we will see what's the difference between alpha keto acid and also the normal amino acid. Okay, why nitrogen needs to be removed? Because based on this reaction that we learned so far, there will be not even one molecule that contains nitrogen. Faham tak apa maksud dia? Anybody of you guys know what it means?
Tak diperlukan.
Maksudnya nitrogen ni tak diperlukan dalam semua reaction yang kita
Terlibat
belajar.
dalam proses-proses
So you understand, right? Maksudnya yang kita belajar so far, glycolysis, Urea Cycle and so on, semua tak ada nitrogen. So nitrogen ni, once our body dah store enough amount in probably muscle, sebab muscle semua perlukan nitrogen kan untuk build up kan, dia akan buang nitrogen ni, tak perlukan dah. Sebab kalau nitrogen ni simpan dalam badan, dia akan hasilkan ammonia, which is poisonous for the brain, which is not good for our body. Okay, so that's why nitrogen need to be constantly being removed from the body if our body have enough storage of nitrogen. Okay, siapa yang tak pernah belajar nitrogen metabolism before? Did you guys ever learn before? Ke ni first time dengar?
First time.
First time so kena detail sikit lah. Okay, so this is important kalau saya tanya exam, saya lupa ni lah. The stages of ammonia excretion. The first thing before we enter into this topic is you need to understand the most important amino acid paling penting dalam semua ialah glutamate. Semua pernah dengar glutamate kan? Pernah tak glutamate?
Nah.
Kat mana selalu dengar glutamate? Dekat mana dengar glutamate selalu? Siapa suka makan nasi goreng?
MSG tu.
MSG, right? Monosodium glutamate, right?
Yeah.
Okay, kan kita tahu our tongue, lidah ni, dia ada banyak reseptor rasa kan? The taste bud. Ada sweet, ada sour, ada bitter. So actually all these taste bud, it correspond to something. For example, we taste sweet because our body needs the glucose. We taste sour because our body needs some kind of pH control. We taste bitter probably because we want to detect the poison. Sebab so usually poison dalam plant ni dia bitter dia pahit. And there will be one taste bud which is umami. Pernah dengar tak? Umami ni quite recent lah bila saintis jumpa. And they found that umami is corresponding to the protein. So another name for umami is actually glutamate lah. Sebab tu MSG ni dihasilkan rasa yang sedap berbanding amino acid lain. Sebab tu kalau kita tambah dalam makanan, kita enhance the taste of umami. So all the food that is high in glutamate, for example meat, cendawan, seaweed, they all contains high amount of glutamate. The initial glutamate that we get in the form of MSG actually extracted from the seaweed, daripada Japanese punya invention lah. But then after some time we found that it is better to produce this glutamate through microbial punya technologies. So what we do is we can actually change the genetic or do genetic modification in the E. coli to ask the E. coli to express more of this glutamate in the media. So kita extract lah, and then kita hasilkan monosodium glutamate. So through fermentation lah, right? So because the glutamate is very important in the body, so that's why our body use glutamate as one of the taste bud in the tongue. Okay, so just side punya information lah, fun fact. So the rule for amino excretion is, kalau amino acid tu bukan glutamate, dia mesti ditukarkan semua kepada glutamate dahulu. Okay, transamination of all amino acid to glutamate if that amino acid is not glutamate. Faham tak the first rule?
Faham.
Okay, so maksudnya kalaulah awak ada amino acid lain, contohnya aspartate ke alanine ke glycine ke all this 19 amino acid ni, ada 20 kan, 19 lain tu kalau dia bukan glutamate, it must be transaminated to glutamate first. Okay, that's the first rule. Transamination means that amine ni ialah group nitrogen kan, betul tak? amine ni? Have you ever heard about the amine group?
Benar.
Okay, so nitrogen lah. Okay, pernah dengar tak bus transnational? Have you ever heard about transnational?
Ya.
Student mesti naik bus kan, mana tahu tapi student sekarang kaya kan. Maybe dia naik kereta, tak tahu lah. Pernah dengar tak transgender?
Nah.
Apa itu trans? Trans ni maksudnya the movement from one to another. Okay, so in this case, it's actually the movement of amine group to another. Okay, so dia transaminate, dia move this nitrogen daripada amino acid ke lain untuk form glutamate. Okay, so that's what we call as transamination. The second rule is after we form the glutamate, we will undergo the process of deamination. Okay, macam tadi saya cakap kalau carbon dioxide tambah panggil carboxylase kan. Kalau kita buang kita panggil decarboxylase, betul tak?
Yep.
Okay, so kalau deamination maksudnya pembuangan nitrogen lah. Okay, so we remove this amine group from glutamate to form what we call as alpha ketoglutarate. Pernah dengar tak perkataan ni? Alpha ketoglutarate ni?
Tak.
Okay.
Benar.
Macam pernah dengar kan? Dekat mana tu?
Krebs cycle.
Okay, Krebs cycle. So kalau tengok ni, nampak tak? Kan kita ada Krebs cycle, acetic acid, isocitrate and so on. Ni ada alpha ketoglutarate. Alpha ketoglutarate ni actually samalah dengan alpha ketoglutarate acid. So actually this glutamate can produce alpha ketoglutarate. Okay, this is what we call semua yang intermediates ni yang kita panggil as alpha keto acid ni yang tadi tu. So actually amino acid is any, kalau kita tambah nitrogen satu dekat sini dia jadilah glutamate. Kalau kita tambah nitrogen satu dekat, for example fumarate, dia akan jadi satu amino acid. Jadi kita tengok dia carbon skeleton dia sama tau. Dia punya rangka tu sama. So kalau kita tambah dia akan jadi amino acid. Nanti kita tengoklah. So nanti bila dah jadi alpha ketoglutarate, kita dah deamine, kita dah buang nitrogen. This nitrogen will be in the form of ammonia that it will enter into the urea. Kita panggil urea cycle lah. Okay, and the third one it will enter into urea cycle lah. So this is the TCA cycle. As you can see, there will be one alpha ketoglutarate here. Kita tengok the first rule dulu. Okay, kan kita cakap the first rule, if that amino acid is not glutamate, dia mesti convert to glutamate first. Ingatkan tadi kan, the first rule tu. Ni. Jadi cakap faham kan yang first rule ni? Faham tak yang first rule ni? Do you understand the first rule? Okay. So I want to show you guys how this transamination occur. Okay, for example, this amino acid, this amino acid is obviously not glutamate lah sebab kan kita cakap nak tukar pada glutamate kan. So this amino acid will contain nitrogen and it will react with alpha ketoglutarate and this nitrogen will be transferred to this alpha keto and it will produce glutamate plus new alpha keto. Siapa yang tak faham? Laju sangat pula kan. Kalau lukis senang ni. Okay, glutamate ada berapa karbon? 1, 2, 3, 4, 5 kan? Betul tak? Glutamate ada 5 karbon kan?
Betul, 5.
Okay, so what happen is alpha ketoglutarate ada berapa? 5 karbon juga 5. Kan? Nampak tak dia actually sama tau alpha keto dengan glutamate ni. Tapi the difference is glutamate ni dia ada nitrogen. So kalau kita tambah nitrogen kat alpha keto dia jadi glutamate. Right. Faham tak itu so far?
Faham.
Okay. Tahu tak pyruvate? Pyruvate ada berapa karbon? 3, right? Kalau kita tambah 1 nitrogen, dia akan jadi new amino acid yang 3 karbon. Amino acid yang 3 karbon ialah alanine. Faham tak?
Apa sekali lagi?
Pyruvate kan ada 3 karbon kan?
Okay.
Okay, kalau kita tambah satu nitrogen, dia akan jadi amino acid, which is alanine. Alanine 3 carbon amino acid. You understand that rule?
Yeah.
Okay, salah satu lagi intermediate anti-acid cycle, fumarate. Dia ada 4 carbon. If we add 1 nitrogen to this fumarate, it will become a new amino acid. Nama dia ialah aspartate. Mana kata aspartate? Pernah dengar tak? Okay, so in conclusion here, if we add the nitrogen to this alpha keto acid, intermediates in the metabolic reaction that we learned before this, it will become a new amino acid depending on the carbon skeleton. Usually the number of carbon skeleton lah. So kita tahu alpha ketoglutarate is 5 carbon. So if you add 1 nitrogen here, it will become glutamate, a new amino acid. Okay, so the first rule is we must convert all amino acid to glutamate. Okay, for example, amino acid yang kita ada kat sini ialah alanine. Okay, saya tulis sini 5 carbon plus alanine. Okay, understand so far yang ni? Kita akan produce apa? Okay, alanine dia akan transfer nitrogen to 5 carbon, right? Ini jadi apa? Glutamate kan? 5 carbon.
Glutamate. Yeah.
Okay. Ni dah hilang 1 nitrogen. Apa jadi to this alanine? Alanine 3 carbon.
Pyruvate.
Dia akan jadi pyruvate. Nampak tak? Faham tak? Siapa yang tak faham? Saya beri satu contoh ni. Katalah kita ada aspartate. We want to convert to glutamate. Okay. So what this aspartate ni 4 carbon Okay. Kita convert kepada glutamate 5 carbon. So kita kena react dengan apa kat sini? Aspartate kan tambah apa? Kalau boleh jawab ni maksudnya faham transamination ni. Aspartate ni dia akan pindahkan nitrogen dia kepada alpha keto kan kat sini kan. So what kind of alpha keto it must react with aspartate to form glutamate?
Alpha keto ada banyak.
Ni alpha keto dah 7 ni. Pyruvate, Acetyl Coa, Acetyl Coa, alpha ketoglutarate, succinic Coa, fumarate and oxalic acetate. Okey, glutamate tadi kan saya cakap dia punya rangka dia apa?
Amino acid campur nitrogen.
Rangka dia alpha keto kan? Glutamate.
Yeah.
So maksudnya ini dia kena react dengan alpha ketoglutarate lah. Faham? So it will produce back glutamate plus apa? 4 carbon alpha keto acid. Fumarate. Kita fumarate lah. You guys understand or not? Okay, satu lagi contoh so you guys can see Okay, kata kita ada ni contoh ni imaginary je lah, 7 carbon amino acid. Okay, kita nak produce sebenarnya tak ada 7 carbon amino acid saya rasa ni contoh je lah. Kita produce glutamate which is 5 carbon. So dia kena react dengan apa? Sini.
Alpha ketoglutarate?
Alpha keto lah dekat sini. Alpha ketoglutarate lah. So dekat sini kita akan produce new alpha keto acid, right? 7 carbon alpha keto acid.
Yeah. 7 carbon.
So you guys understand so far the transamination?
Doktor,
Faham?
Okay.
kenapa kena ambil alpha keto?
Kenapa kena ambil alpha keto? Sebab
Sebab glutamate tu punya rangka is 5 carbon.
yes.
Oh, so maksudnya apa yang kita nak react tu kita kena tengok dia punya hasil tu ke?
Dia punya summary yang paling senang ialah the summary is every amino acid must react with alpha ketoglutarate in order to produce glutamate. Make sense tak?
Makes sense.
Sebab alpha ketoglutarate dia yang can produce glutamate. Make sense tak? Siapa yang tanya tadi?
Saya.
Okay. So maksudnya 19 amino acid yang lain kan ada 20 kan? Must react with alpha ketoglutarate in order to produce glutamate.
Glutamate.
So the first rule is every amino acid must be converted to glutamate first by reacting with alpha ketoglutarate. Okay, and the second rule, ingat tak tadi? Cakap deamination means the removal of amine group.
Nitrogen.
Okay, ni jadi dalam liver cell juga. What happen if we potong one nitrogen from here? Kita akan produce balik alpha keto lah, right or not?
Yeah.
Betul.
And one ammonia. This ammonia can enter into urea cycle. What happens to this alpha ketoglutarate?
Repeat.
Repeat balik, masuk balik dalam Celsius cycle. Understand?
So
Faham.
proses ni nak buang amonia tu lah.
Proses nak buang ammonia je.
Okay.
Sebab kan kita cakap tadi kalau kita ada lebih, katalah ada amino acid lain, glycine lah, aspartate lah yang terlebih dalam badan, kita nak buang dia punya nak buang dia punya nitrogen tu. So everything must be converted first to glutamate before this glutamate can go through deamination to remove this ammonia. Okay? So kalau faham dia agak straightforward lah. Okay, kalau lukis kat whiteboard is actually easier for you to understand. But I can see some of you guys understand already, so you guys can probably ask your friends or can ask me directly if you don't understand. This is the example you can work with on later on, just to make sure that you understand the process of transamination and also deamination. So I just draw you this funnel. So all amino acid must react with alpha ketoglutarate in order to produce glutamate before this glutamate can produce ammonia through deamination. Okay, again, same. Okay, same. I just want to show you guys that the name of the enzyme that is responsible for the transamination usually based on the name of the amino acid that is here at the substrate. For example, if alanine react with alpha ketoglutarate, we will call this enzyme as alanine transaminase. Okay. For example, if here is aspartate, we call as aspartate aminotransferase or transaminase. Okay, that's nothing lah. You don't have to remember the name of the enzyme. Okay, so this is the importance of glutamate. Like I told you before, the various aminotransferases in the liver all funnel excess nitrogen to glutamate. 50% converted to aspartate and 50% deaminated into alpha ketoglutarate. We will see how aspartate is important in the reaction as well later on. Glutamate can be deaminated by glutamate D Hydrogenase to give ammonia, contributing up to 50% of the nitrogen in the urea cycle. Another half is contributed by aspartate directly. This permits a combined nitrogen excretion pathway described as trans-deamination. So this one is actually transamination and deamination, tapi dia gabung dia jadi trans-deamination. This process demonstrates the central roles of glutamate in the overall control of nitrogen metabolism. So that is why we have a receptor for glutamate in our tongue, because glutamate is very important in the body. Glutamate can also be converted to glutamine, another kind of amino acid. Glutamate ni dia ada satu nitrogen je, tapi glutamate ni dia ada dua. That's why it can act as a storage of nitrogen. Okay, and one of the primary function is to transport ammonia in the blood lah. For example, if you form glutamine in the muscle cell, it can be used to transport in the form of glutamine into the liver for the process of excretion of ammonia. Okay. Kalau tengok sini ada satu je nitrogen in glutamate, tapi ammonia ada dua. One here and another. Glutamine ada dua. Satu sini, satu sini. Okay. So glutamine is the product after the glutamate lah if we add another ammonia. And alpha ketoglutarate is the carbon skeleton of both glutamate and glutamine. Okay. Ammonia. Every amino acid contains at least one amino group that need to be excreted in the urine. Amino acid catabolism generates ammonia which is sensitive to the brain tissue. So we need to excrete this one out. Some amino acid is stored in the muscle. So we have some kind of storage in our body. But if we exceed this storage, we need to excrete them out. Cell get rid of the excess ammonia by transamination and deamination. In other tissue, ammonia is captured by alpha ketoglutarate to form glutamate. This glutamate captures another ammonia to form glutamine before transported to the blood, in the blood to the liver. Glutamate is also a neurotransmitter. Glutamate is also precursor to GABA, which is another important neurotransmitter. Pernah dengar tak GABA ni? Pernah dengar tak? Siapa yang tak pernah dengar GABA? Gamma aminobutyric acid. Kalau siapa suka makan fermented food, especially Japanese food, dia banyaklah GABA ni. Pernah dengar natto tak?
Pernah.
Okay, natto tu kan dia macam slimy tu kan?
Yeah.
So it actually high in this neurotransmitter lah GABA. So it says GABA is able to help you in many things. For example, help you in your sleep, to sleep faster and so on. Dia macam hormon neurotransmitter yang bagi you penenang. So GABA ni, the precursor is glutamate. So if you have too many ammonia in the body, this ammonia can come and bind to glutamate and form glutamine. So bila glutamate tak cukup lah untuk buat GABA. So orang yang ada something wrong in the metabolism of nitrogen can have lower amount of GABA in the body. Okay, so that's why high concentration of ammonia defeat the concentration of glutamate because it will produce glutamine, and it is also toxic to brain cell as well. Okay, so what happened is this ammonia will undergo the process we call as urea cycle to excrete this urea. Okay, kita tengok sikit, tak payah ingat semua, cuma kena ingat urea cycle happens, the first one is in the liver cell. Dulu saya pun ingat kat kidney, tapi actually dia banyak happens kat liver cell. Lepas tu baru urea ni transported in the blood to the kidney to be excreted. So yang kuning ni comes from deamination just now. Yang kita tengok glutamate tu. Okay, and it will go around and form this carbonyl phosphate, and then it will go out from this mitochondria in the liver cell into the cytoplasm. And citrulline, you will form the citrulline which contains one of the nitrogen or ammonia just now yang kita deaminated from the glutamate tu. Lepas tu aspartate akan datang dan dia akan donate one of the nitrogen to this molecule. So kita sekarang ada dua nitrogen. Boleh nampak tak kuning dengan purple ni? Kuning dan purple ni, nampak tak? Saya cakap pay attention to yellow and purple boxes.
Nampak.
Okay, so kan tadi saya cakap 50% of the ammonia ataupun nitrogen comes from glutamate, another 50% comes from aspartate. So inilah prosesnya, 50% satu ammonia datang daripada glutamate, another one ammonia comes from aspartate. So 50% each lah. So now kita ada this molecule that contains two nitrogen. And then this molecule will break down into two components, which is fumarate and arginine. Ingat tak tadi saya cakap aspartate ni, dia boleh produce fumarate. Nampak tak molekul aspartate ni? Okay, 1, 2, 3, 4 kan? Ni fumarate ni 4 juga kan? Tapi tak ada nitrogen. Right? So sebenarnya dia bergabung dengan citrulline ni, dia pecah balik je. dia pecah balik jadi fumarate, tapi dua nitrogen ni dia tertinggal ke arginine. Faham tak? Understand? Do you understand? Dia gabung lepas tu dia pecah balik. But when they break again, this one becomes fumarate, this one becomes arginine. In fact, arginine is one of the 20 amino acid. So this arginine will cut this one and form urea. Ni urea punya komponen adalah C double bond O and ada dua nitrogen. Ada dua ammonia. Okay? And this arginine bila dah potong dia jadi balik this component of ornithine. Ornithine ni salah satu jenis amino acid lah, tapi bukan 20 amino acid. Dia ada dua jenis amino acid. Satu yang 20 itu we call as protein amino acid. Yang bukan 20 itu dia panggil non-protein amino acid lah. Dia tak menghasilkan protein. Tapi tak penting untuk tahulah. So kita tengok eventually it will produce urea that can be excreted in the urine. And this ornithine can enter back into the mitochondria and repeat the process of the urea cycle. Ingat tak saya pernah cakap urea cycle actually was found by the same person, Crab. Ingat tak?
Ingat.
Okey, kejap ada telefon pula. Ya. Ada tanya? Rasa saya cancel. Oh, sebab saya pun boleh ingat apa saya tadi biasa saya meeting. Okay, can we continue?
Yes.
Okay, so now we already produce this urea, and this urea will be excreted out. Okay, and then we complete lah the nitrogen metabolism. Okay, this is just we were talking about the crab, right? Okay, the person who found this one also found TCA cycle, so that's why we call that TCA cycle as crab cycle. But this one we call as urea cycle lah. Tapi TCA cycle ni dia link dengan fumarate, because if we remember, fumarate is one of the intermediate of TCA cycle. So that's why we call this one as crab bicycle. Dia ada dua cycle, bicycle lah. Alright, so that's just additional information for you to know. You don't have to remember, just to show you that the same person found the cycle. And this one, crab bicycle, urea cycle is actually the link of fumarate. Okay, from arginous succinate just now. Okay, so I just want to say in this slide is all amino acid are metabolized, resulting in the intermediates which are central to either carbohydrate, glucogenic, or lipid ketogenic metabolism. Some amino acid yield both kind of intermediate. You can see it from this figure lah. Okay, any amino acid that can produce acetyl CoA or acetyl CoA, the product of acetyl CoA, we call as ketogenic amino acid. But if it produce other than these two, for example, it can produce pyruvate, okay? We can produce oxalic acetate, it can produce fumarate. Nampak tak ada aspartate kan, dia boleh produce fumarate kan? Alpha ketoglutarate. Tadi kita tengok glutamate and glutamine can produce alpha keto. These all we call as glucogenic amino acid. Ingat kan gluconeogenesis? Ingat tak gluconeogenesis?
Yes.
So what happen is, kan kita cakap gluconeogenesis will happens bila pyruvate form glucose, right? Or oxalic acetate form glucose. Ingat tak? Pyruvate kan boleh form glucose, right?
Boleh.
Okay, and we also established that oxalic acetate can also produce glucose. So if the amino acid can produce any of the intermediate in the TCA cycle, it can go round and eventually produce back the oxalic acetate. Faham tak? Katalah glutamate ni boleh produce alpha ketoglutarate, alpha keto ni boleh produce oxalic acetate kan? Sebab dia boleh pusing sini. Okay? So dia boleh produce glucose. That's why we call it as glucogenic amino acid. And
maksudnya glucose yang from the after process of, maksudnya daripada amino acid dah.
Glucose ni dia form daripada amino acid ni. Amino acid ni dia boleh produce glucose. The easier way to say it lah. Awak faham kan?
Faham.
Okay, so it means then from arginine, okay, for example, from arginine or proline, we actually can use this amino acid to produce glucose, okay, by the liver. But if this amino acid produce acetyl CoA, dia tak boleh produce glucose, dia will produce something else, which is ketone bodies. Ingat tak kita belajar acetyl CoA ni boleh produce ketone bodies?
Ya.
Okay, so kalau dia boleh produce acetyl CoA, dia produce ketone bodies instead. It will not produce glucose. But as you can see in this figure, some of this amino acid, it can produce both ketone bodies and glucose. For example, tryptophan. Sini ada tryptophan, right? Biru. Ni pun ada tryptophan juga, warna merah. Sebab dia boleh produce both of these intermediate. So dia boleh produce ketone bodies, dia juga boleh produce glucose. Okay, that's all. Any question before we finish today? Next week kita buat test 2 boleh tak?
Objektif ke subjektif, Doktor?
Semua subjektif.
Pendek-pendek je jawapan.
Pendek je, senang je. Semua kalau faham tu boleh jawab je. Okay.
Bab berapa?
Tengok balik video-video kan akademi. Banyaklah yang senang kan untuk fahamkan berbanding saya bagi lecture. Okay. So kalau siapa yang nak study extra, bolehlah datang ke. So you guys are in the faculty right now, right?
Yes.
Okay. You guys can come. Or maybe we can play a quiz. Just tengok Kahoot ada tak? Nak play a quiz tak? Just as a revision for next week.
Boleh.
Sekejap. Dah lama dah quiz ni, saya tak ingat dah. Oh ini. Tak ada pun. Maybe email nama. Okay, can you guys see the game pin? Tahu tak macam nak guna Kahoot ni?
Oh.
Okay, attendance nanti saya tengok. Saya maybe lupa kot. Ni player lama ni rasanya. Okay, so okay, can we start now? So because this is revision, I haven't had the opportunity to take a look at the question yet. Okay, so maybe some are correct, some are not lah. Okay, but we just play for fun. This one. Okay, so we start. Lamanya satu ni. Okay. Okay, so this is the correct answer. Which one is metabolic pathway? Cyclic divergent. Reverse is not metabolic pathway that we learn. Kita ada belajar cyclic divergent, convergent, spiral, okay, and so on, but not these two. Okay. Second one. Okay, so this is pretty straightforward. Okay, so because we add H2O, but no breaking down is happening, so this one is non-hydrolytic reaction, not hydrolysis. Okay. Okay, so if you remember the gluconeogenesis, we cut the phosphate so that the glucose can leave the cell. Okay, so to stabilize the blood glucose level. So if we attach the phosphate, means that we trap the glucose inside the cell. Okay, CK. I'm not sure you are from this cohort or not. Okay, this is the only non-characteristic of the regulated reaction, okay, in glycolysis, in TCA cycle, and so on. Okay, the yeast in the bread, the final product is actually ethanol. So we Muslim can consume the bread because the ethanol will be burned off during the baking of the bread. Okay, because red blood cell doesn't have any organelle, means that they don't have any mitochondria, means that they cannot conduct the aerobic respiration, so they need to conduct glycolysis only. So red blood cell always produce lactic acid as their byproduct. Okay, ni ramai tak ada jawab. Quite many of you guys don't manage to finish this question. If you remember the breakdown of glycogen, actually they skip the first process of glycolysis. The first process of glycolysis requires 1 ATP, right? The investment phase. So if they skip this one, means that they can skip one of the investment phase. So they can produce more ATP. I mentioned this one before, if you remember. Okay, so glycogen storage disease will cause weakness. Okay, there are two answers in this question because there is not enough glucose in the blood. Second, protruding liver because a lot of glycogen actually accumulated in the liver, so it will protrude from the chest. Nama jawab diabetes. Okay, most of you guys got this one correct. Okay, the blue one is actually the process in diabetes type 1, not type 2. Okay, most of you guys get this one correct. Kidney is correct as well because like I told you guys, kidney is always a sister to liver. Most of the processes in liver can happen in the kidney as well, but in a smaller amount. Okay, so anybody who choose kidney is correct as well. Okay, actually the red and the yellow one is kind of correct as well, but the main importance is to balance the NAD plus and NADH ratio because this NADH cannot enter into electron transport chain, so it must be converted through some other means. In this context, it must be converted to lactic acid, so it can convert back NADH to NAD plus. Okay, actually the most accurate one is Acetyl Coa because this one is very important for the TCA cycle. But this process also produce NADH and CO2. Okay, the production of Acetyl Coa from pyruvate. Banyak lagi ni. This one is the last question. Okay, the two main major purpose of TCA cycle is it can become, or what we call the reaction is amphibolic. Means it can supply precursor for other biosynthetic reaction in anabolic reaction, or it can also produce NADH for the production of ATP. Can we stop here? Kena, teruskan.
Yes.
Stop
Stop.
Yes, stop.
Baru half soalan ni, jadi ada lagi 14 ni. Pasal ada banyak quiz lagi lah. So if you guys can answer this one, actually you understand everything that I taught you so far. But maybe next time we can play together. Okay, so that's all. Any question before we finish? Nanti saya buat attendance after this. I will open the attendance until tonight lah. Okay, because I probably forgot. So I will see you guys next week for the test 2. Okay? And this one will be the last lecture. After this, you will be having lecture with Dr. Anip on the DNA and probably photosynthesis. I'm not sure about the photosynthesis. You need to ask him. Okay, thank you. Bye.
Thank you so much.
Thank you, Doctor.
Thank you, Doctor.
Thank you, Doktor.
Bye.
Thank you, Doctor.